Hi, my name is Chris Burns, the CEO and Managing Director of Amplia Therapeutics. Amplia is an Australian biotechnology company listed on the ASX. And today, I'm very pleased to take you through our slide deck that introduces the company and our programs. So, Amplia Therapeutics is developing a series of compounds that target focal adhesion kinase, or FAK. And the drugs are being developed in the fields of oncology and in fibrotic disease. The run through today will cover a corporate overview, some background about the drug and FAK itself, some preclinical data showing the potential of narmefotinib in the treatment of solid tumours and why we believe it's a best-in-class agent. Following on from that, we'll talk about the clinical data we've obtained to date and finish up with opportunities in fibrotic diseases as well, including some of our preclinical data in that area. And then finally, what our plans are for the calendar year 2024. So by way of corporate overview, the company, as I noted, is listed on the Australian Stock Exchange, the ticker code ATX. We're headquartered in Melbourne, which is in the southeast corner of Australia, and we have a market capitalisation of about 16 million Australian dollars. Within the Australian context, we have some of the best known institutional investors in the biotech space: Platinum, Blue Flag and Acorn Capital. And we have an experienced board with international experience in drug discovery, development, and in governance and legal matters. By way of a rapid summary, we are developing the best-in-class FAK inhibitor called narmefotinib for cancer and fibrotic indications. Our primary focus has been cancer, and in particular pancreatic cancer, and we have a current trial underway in Australia and South Korea. We expect an interim readout on that trial in mid-2024. We have a small team, but an experienced team of drug developers, and we work with a network of exceptional contractors and consultants in science, clinical science, and regulatory matters. And those consultants are based around the world. Some key points: we have an open IND for narmefotinib to run the clinical trial in pancreatic cancer in the US. We have orphan drug designation for both pancreatic cancer and IPF, pulmonary fibrosis. And we have a lot of really compelling preclinical data that I will present some of in models of pancreatic cancer, as well as demonstrated activity in models of ovarian cancer and IPF that I'll touch on today. A pipeline is shown here. We've completed a healthy volunteer study. I'll speak to that. The pancreatic cancer trial called ACCENT is underway. We've got some really exciting early data in ovarian cancer, and we want to move that forward very quickly over the year. And we've got some earlier work in solid tumours and, as I said, IPF. Our second drug, called AMP886, is not as well developed, and we'll do some background work again over the year to explore additional opportunities for that agent. So a little bit about FAK and narmefotinib itself. So FAK is a critical signalling molecule that's overexpressed in a number of cancers, pancreatic cancer and ovarian cancer being two of those, where in those cancers it drives things like proliferation, survival, cell migration, but also drug resistance. So it's a key resistance pathway for other agents. It's upregulated under cell stress, so on the treatment of those cells or cancers with chemotherapies, you see FAC levels increase to compensate as a cell stress response. All those effects are within the cancer cell itself, so the cell intrinsic effects, but there's also cell extrinsic effects where we're looking at the role of FAC in tumor fibrosis, so the fibrotic scar tissue that forms around a cancer lesion. Also, an immunosuppressive environment that's present in a lot of solid tumors where the immune system doesn't work well against the cancers. And lastly, FAC is also involved in angiogenesis, so the feeding of blood supply to tumors. So by blocking FAC, by blocking the activity of this particular protein, we have a way of targeting processes that are happening within the cancer cell and around the cancer as well. So if you like, it's a double whammy to affect those cancers. In fibrotic disease, we're really focused on these issues of fibrosis and angiogenesis. So by targeting FAC, we're having a beneficial effect on decreasing the fibrosis that's occurring in those diseases. And I'll speak a little more to that in a minute. There's opportunities to combine FAC with a range of FAC inhibitors with a range of drugs that are used currently or that are in development. FAC inhibitors have potential to be used as single agent drugs in non-oncology indications, but in oncology, we believe the best opportunities are when combined with other drugs. Some targeted agents are mentioned here that are in development by, or actually already approved by pharma and biotech. Particularly note KRAS inhibitors. That's an area of considerable interest these days. We've got 2 KRAS inhibitors approved for the treatment of lung cancer, and a lot of development happening in the industry for inhibiting this particular oncogene in a range of other cancers. What's known, though, is that there's resistance that develops to those KRAS inhibitors. And there is strong data, and we believe, based on our own studies, that by inhibiting FAC in conjunction with these agents, and particularly KRAS inhibitors, there's great potential to enhance the efficacy of those targeted agents. There's also opportunities and strong data in the scientific literature showing combination of FAC inhibitors with immune checkpoint inhibitors, things like PD-1 and PD-L1 inhibitors that are now approved and have been very successful drugs. And this is a signaling pathway showing within a cell that FAC sort of sits above, if you like, a lot of well-known oncogene or oncogenic programs, so that drive invasion, metastasis, block apoptosis, and increase survival and growth. And therefore, by blocking FAC in conjunction with other targets within this network of signaling events, that you will have added, possibly synergistic effects. And this is data, not our own data, but published data showing that a combination of a FAC inhibitor and a KRAS inhibitor in a mouse model of KRAS mutant lung cancer, that the combination is much more effective than the individual agents alone, blocking tumor growth and extending survival. So again, speaking to the potential here in KRAS inhibitors. So a little bit about the molecule. The drug is a small molecule, meaning it can be taken by mouth. We currently present the drug as a capsule. It is highly potent and selective for FAC. That's what this image shows here. Basically, of all the kinases that are present in the human genome, so it's about 530, we've screened our drug against about 430 of those kinases, and we inhibit predominantly only FAC. There's a little bit of off-target activity, but nothing significant. So that's highly selective. It's also highly potent. It has nanomolar potency. It has a strong binding affinity to FAC, which is shown here. This is the crystal structure of the compound inhibiting the protein. There are, importantly, no flags and no observation of any drug-drug interaction showing that our drug can be used in combination with other drugs. Its pharmacokinetics are such that the drug is given once a day. It has no food effect either, so you can take it with or without food. And that combined data set alone sets us apart and better than the competitive agents, of which there are 2 currently being developed, which is what I show on this slide. So namafotinib, highly selective, excellent pharmacokinetics, no drug-drug interaction issues that we've seen. And to date, from our clinical studies, the drug has been safe and well tolerated. Defactinib, which is the most advanced compound being developed by US company Veristem, is not as selective. It's a drug that has to be given twice a day. There is evidence of some drug-drug interaction issues with defactinib. Having said that, that drug has shown some promising data in low-grade serous ovarian cancer, if you like, helping validate the role of FAC in disease. The other drug being developed by a Chinese company is ifabemtenib. That drug has reasonable selectivity and a good PK profile, but there are off-target activities that are observed in some clinical data that's been reported to date. And the 4th compound I wanted to mention, which is no longer in development, but that is a compound from GSK, has real issues with both pharmacokinetics and drug-drug interaction issues, which is one reason why the drug was discontinued, as far as we're aware. In terms of intellectual property, our drug was invented here in Australia in 2000, and the patent was filed in 2012. Since then, we've looked at evergreening that IP with salt form and additional patents around the drug that is being currently developed, as well as method of use patents, which have been filed from 2021, and others are in filing as we speak. So very focused on life cycle management, and worth noting again that we do have orphan drug designation, which of course allows us to extend the commercial opportunity for the drugs. So in terms of solid tumours, we have been working with a number of academic labs to demonstrate the efficacy of the drug in models of cancer. And the work shown on this slide here, it's quite a busy slide, but basically it shows that in the test tube, as you increase the concentration of our drug, you can block the formation of the deposition of collagen and the formation of fibrotic tissue from fibroblasts that they're growing in the test tube. So this is a very simplified representation of the process that happens in solid tumours when you get that scar tissue forming around the tumour. Our drug blocks the formation, the first step in the formation of that scar tissue. No drug, brightly pink. As you increase the concentration of drug, less pink, less of that scar tissue. And that's shown here as well with a different kind of imaging format here. As you increase the concentration of drug, you get less of that complex collagen forming. And importantly, we've gone and repeated that in an animal model where we've actually, under the skin, introduced a pancreatic cancer tumour, which itself is fibrotic. And you can measure that level of fibrosis, again, using that sort of pink staining I showed you, or the purple staining before. And in the presence of our drug, dosed orally to the mouse, and this is a short-term assay, only over 4 days, you can see a significant reduction in that amount of collagen and cross-linked collagen. And that corresponds with a decrease in the amount of active FAK in those tumours as well. So correlating the activity we're seeing against the fibrosis, against the activity we know is present against the FAK protein. In a separate model, this time a model where there's an intact immune system in the mouse. Sometimes the models are run without an intact immune system. But this is a mouse with an intact immune system. And what we've shown here, if you look at a tumour environment after treatment with our drug for 12 days, you can see a significant reduction in macrophages, so a significant reduction in some of the cells that actually decrease the immune response in that tumour. So a decrease in macrophages and particular subsets of macrophages, and importantly, an increase in CD8 effector T cells. So this is the T cells that actually try and kill the cancer alone. So in terms of immuno-oncology, we say we have a decrease in tumour-associated macrophages, a decrease in myeloid-derived suppressive cells, and an increase in CD8 effector T cells. So these are all the kind of things you want to see that actually enhance the potential of the immune system to work against the tumour, and also enhance the potential of immune checkpoint inhibitors to work. As I mentioned, FAK is overexpressed in a number of cancers. So ovarian cancer shown here, where you see high overexpression, pancreatic cancer here, but other cancers such as uterine carcinoma, head and neck cancer, and so on and so forth. Importantly, there's clear genetic data that shows that in humans, where patients have a naturally lower level of activated FAK in the fibroblasts around the tissue, they have a much better survival, much better outcome over time compared to patients that have highly active FAK in their tumour environment. Those patients, unfortunately, have a much worse outcome. This is in pancreatic cancer, but this data has been reflected in a number of different cancers as well. So again, genetic support that targeting FAK actually will have a beneficial effect. This is some data we've done with an academic group here in Australia, one of the top medical research institutes called the Garvan Institute. And we're looking at a survival graph here of mice that are given pancreatic cancer, a particular type of pancreatic cancer, in this case, one that's been actually taken from the patient. And if you treat that mouse with a placebo or with vehicle, if you like, the mice succumb to the disease quite quickly in about 75 days. And if you give them, we look at this lighter green line here, gemcitabine Abraxane, which is one of the standards of care for pancreatic cancer, you can see that prolongs life considerably. However, if you add our drug, narmefotinib, on top of gemcitabine Abraxane, so you give it prior to giving gemcitabine Abraxane, you can prolong survival significantly by about 35%. And in fact, this survival plot probably underestimates the effect of the drug because as a consequence of the way the experiment's done, some of the mice are removed. They haven't died. They're removed simply because of an ethical issue to do with the gemcitabine Abraxane dosing. We're looking here at the tumour size from that very experiment. And this is what you really see. At about day 50, day 55, the mice getting gemcitabine Abraxane the tumours continue to grow in size, whereas the mice getting the AMP945, it continues to level off. And we've taken this out now for a number of days beyond here, and the tumour size does not continue to grow on average. And that's correlated with the decrease in proliferation signals and an increase in cell death that we see in those cancer cells. That's looking at a combination of narmefotinib with gemcitabine Abraxane. The standard of care for pancreatic cancer in the US is a drug combination called FOLFIRINOX. And again, FOLFIRINOX does prolong life in this mouse model of cancer compared to vehicle-treated animals alone. But when you add narmefotinib, in addition to FOLFIRINOX, again, we see this extension of life of about 30 to 35%. So we think these two bits of data here, but we have other experiments in mouse models that I'm not showing that show the same effect. That combining narmefotinib with the standard chemotherapies in pancreatic cancer, gemcitabine and Abraxane, or FOLFIRINOX, has a significant extension of life in those mice. I'm shifting gear now to ovarian cancer, where we're really excited about some opportunities that are developing in that space. FAK is known to be involved in ovarian cancer. As I showed you before, there's an increase in FAK levels in ovarian cancer compared to normal tissue, but you particularly see an increase after treatment with chemotherapy and particularly around cancer stem cells, so a kind of a persistent cancer cell where you see very high FAC levels. And this is work that's been published by a collaborator and now a consultant of ours, Professor Dwayne Stupak, I should say, at the University of California in San Diego, which shows that you see high FAC levels compared to normal ovarian tissue. You see high FAC levels in cancer cells, particularly high after treatment with chemotherapy, which again is reflected here. And it's also higher FAC levels as the disease progresses. So very consistent with what I showed on the previous slide. In ovarian cancer, FAC is really one of the key proteins that's driving disease. So working with Dwayne and with Professor Stupak in a mouse model of ovarian cancer, we can show that the cancer after the initial treatment, which is what happens in the clinic with patients, they put on a maintenance therapy in the hope that after decreasing their tumour size, the maintenance therapy will keep that tumour in check. That doesn't always happen, and tumours slowly gain resistance. And what is shown here is that placebo or vehicle-treated animals, the tumour regrows quite quickly. But even using the standard of care, which is what's called a PARP inhibitor, the tumours can recover over time. Tumours treated with the FAC inhibitor actually don't show any or very, very minimal growth over time. So there's a real opportunity demonstrated in this data here of FAC inhibitors in the treatment of ovarian cancer, and particularly ovarian cancer that's resistant to other treatments. We are currently speaking with a number of clinicians about how to move this forward into the clinic. So a summary of all that work in solid tumours is this, that we have a direct effect on the tumour microenvironment, the TME. We reduce fibrosis and we improve the immunoreactivity of the microenvironment. In addition, the drug has a direct effect on the tumour itself around proliferation and survival. We've shown that the drug enhances chemotherapy and it sustains that chemotherapy response to resistance pathways. We've got data that I'm not showing you here, other than the pancreatic and ovarian data. We've got data in other cancer types as well, showing opportunities more broadly outside of cancer and ovarian. And the recent data from our competitors, both Verastem and Inxmed, in ovarian cancer supports the potential for FAC inhibitors to enhance the activity of chemotherapies in those diseases. I wanted to note that Professor Stupak made this comment early on in our interaction that he had looked at all FAC inhibitors in his hands. The Amplia drug, namafotinib, is far superior in terms of both activity and tolerability. So moving to the data we've seen in the clinic, namafotinib has been in a healthy volunteer study. The potential here is to consider the drug both in oncology and non-oncology indications. So we wanted to do a trial in healthy volunteers and look at the safety, tolerability, and pharmacokinetics of the drug. And what we showed here, we dosed 56 patients over a number of different dose levels. We showed that the drug was safe and well tolerated. There were no serious adverse events, no withdrawals from that trial, and no safety trends observed at all. Importantly, the drug showed once-a-day pharmacokinetics. So that's what's shown here. Over 24 hours after dosing, you can see the drug reaches maximum concentration and then is cleared over time. And what we've found in the clinic on multiple ascending, after multiple dosing of the drug, that we reach a steady state after about 4 days. Drug levels increase in a linear manner as you increase the dose. And what's shown down here is we were able to take skin punch biopsies of these patients and look at the level of activated FAC in the skin after dosing with the drug. And what you can see, while the data bounces around a little bit here, as you increase the concentration of the drug, you actually decrease the amount of that active focal adhesion kinase, showing that not only do we see an increase in drug levels, but we see a decrease in the active FAC as well, showing that there's this pharmacokinetic and pharmacodynamic relationship of the drug. Our clinical trial in pancreatic cancer, once we'd shown the good pharmacokinetics and the safety, was initiated in 2022 in Australia. This is a first-line therapy and targeting patients who are non-resectable or metastatic, so particularly advanced disease, but first-line patients. And the way we run the trial is to pre-dose with namafotinib prior to giving on a weekly cycle the gemcitabine and Abraxane. So over 1 month, patients get 3 doses of gemcitabine Abraxane, and prior to those 3 doses of gemcitabine Abraxane, they take namafotinib as a pill in the lead up to those doses. We elected to follow this approach for 2 reasons. One, this is how the animal experiments were run that showed the excellent activity. But also we felt that the burden on these patients who were quite unwell, the pill burden and burden for the liver to deal with additional drugs would be taken away if we only dosed in the lead up to gemcitabine Abraxane. Gave the patients a day off while they recovered from the chemotherapies, and then came back to namafotinib. So that's what we've done. And what I can tell you is that we completed the phase 1b portion in November of last year. That was through the sites in Australia. There was really promising data that came out of that. The drug was safe and well tolerated in those cancer patients. And all patients who came onto the trial stayed on the trial. They could have elected to come off the trial after the 28 days. All patients stayed on that trial. We've had patients stay on the trial now for getting up to 10 months, which is fantastic. Many of these patients would normally succumb to disease within about 4 to 5 months. And we've had, of the 14 patients that were in this phase 1b trial, we've had the majority have stayed on trial for more than 5 months. I'll show you a little bit of efficacy data in a minute. We've now initiated the phase 2a portion of that trial, where we will recruit up to 26 patients in both Australia, and we've recently opened sites in South Korea after receiving approval from the Korean regulator. What we're looking for at this point is an objective response rate and other safety criteria. And particularly, we want to see 6 or more patients having a confirmed partial response. If we see that, that will be promising data and compare very favourably with historical data of patients in pancreatic cancer treated with gemcitabine Abraxane alone. So 6 or more patients to receive a partial response. Then we'll continue on with the trial, enroll another 24 patients. So there'll be 50 patients in all in this phase 2 trial, and we'll be looking at objective response rate, duration of response as the key readouts, and looking at some other markers as well, overall survival and also some biomarkers. So what did we see to date? Well, we looked at 3 different dosing cohorts, 100, 200, and 400 milligram doses. What we saw is the drug was safe and well tolerated, as mentioned. There was 1 dose-limiting toxicity, which was an uncontrolled nausea that led to us expanding that particular cohort. That was the 400 mg cohort. But really the only adverse event that we think relates to the drug is a grade 3 or below toxicity that was seen, fatigue, I should say, that was seen in more than 1 patient. So some fatigue possibly caused by our drug. remembering, of course, that these patients are receiving cytotoxic gemcitabine and Abraxane and already unwell. In terms of efficacy, we wanted to show some signs of efficacy, but it's worth noting that this combines only 14 patients, so it's a small patient number, and it includes patients on the lower 100 milligram dose as well as 200 mg and 400 mg doses. So I want to stress this is not power for efficacy. As I said, most of our patients, 9 of 14, have stayed on drug for more than 5 months. We've not seen any complete responses, but we did see 6 partial responses and 7 stable disease. So 13 out of 14 patients show a disease control rate. That compares very favourably with historical data for gemcitabine Abraxane alone. So while early, the trend is definitely positive for efficacy for this molecule and gives us hope as we move forward into this phase 2 trial that's underway. As we finish up, I wanted to switch to fibrotic diseases and just share a little bit of data in a model of IPF. So this is the industry standard model of idiopathic pulmonary fibrosis, where mice are challenged with a toxin to induce a fibrotic response in their lung. And what you can see is if you image the lung, a healthy lung, you don't see much collagen. You treat them with that toxin, bleomycin, and you see a lot of collagen stained purple here. You treat those animals with our drug, and that most of that collagen disappears, so that the lungs look much more like a healthy lung. We've repeated that both at an academic lab and at a contract research organisation. This is the more recent data that I wanted to show. So you can see this is a measure of lung fibrosis. You can see the vehicle-treated animals or placebo-treated animals get very high fibrosis. That's decreased on treatment with AMP945 or narmefotinib, as we'd call it now. And it's decreased to the same level as what we see with Ofev or nintedanib, which is the standard of care agent. So just as good as standard of care agent, but our drug is much better tolerated. The standard of care agent still, while being a very successful drug, is actually not well tolerated by patients. There was no combination effect of those two drugs, probably indicating that that's the maximum effect you can see in this assay, in this experiment. We have data in a model of non-alcoholic steatohepatitis, but I don't want to elaborate on that. That's not an area we're currently focusing on. But it does actually underscore the activity of both narmefotinib and our second agent, AMP886, to impact pathological fibrosis in a disease model. So we really think this data underpins the potential of these agents in non-oncology but fibrotic diseases. And it's as good as the standard used in these studies. So finishing up our plans going forward, as I mentioned, we have Korean regulatory filing, we have a US IND, and we hope to initiate a trial in pancreatic cancer in the US sometime in the second half of 2024, where we're combining narmefotinib with FOLFIRINOX, which is the preferred treatment in the US. We will get an ACCENT readout mid-year from our current trial, where we're combining with gemcitabine Abraxane. That should be on or around the middle of the year. And we want to continue to expand the therapeutic opportunities, and particularly ovarian cancer, we feel is a really exciting area to be in. Clearly unmet medical need in that disease, and we have promising preclinical data as shown. The final readout from the ACCENT trial will be in the early part of 2025, based on the recruitment rates we currently see. So we're looking for opportunities to invest to support these additional clinical trial activities and to continue the ACCENT trial. And that investment will be up to $50 million. But we're also looking at some regional licensing opportunities in the shorter term, and speaking to pharma and biotech companies at the moment, and there's increasing interest in the FAK inhibitor space. And so with that, I will finish the talk and thank you for your attention.